Okay, so welcome to topic 9, part 2 of this week. This will be covering videos 3 and 4. I'm George, my email's down here. So firstly, we'll be going over glycolysis. So there are two phases of glycolysis. There's an energy investment phase and an energy payout phase. So two ATP molecules are used during investment while 4 are produced during payout. So more come out from payout than you invest, which is important because otherwise the cell wouldn't do it if you get less out of it. So glycolysis is a series of chemical reactions, as you can see here with like the graphic. There's like yeah, there's a lot of reactions here. You'll need to memorize like each reaction, or well, you'll need to memorize like each enzyme that like catalyzes the reaction. So like this first reaction where glucose goes to like glucose 6-phosphate. You just need to know that like what happens is that a phosphate group is added onto glucose. I don't think you'll ever be asked like to like know like what the name of the enzyme that did it is. Just know that like a phosphate is added to glucose, then like another one is added, and like what that does. So I'll be going over that right now. So like, the first two steps of glycolysis requires ATP. So phosphorylating glucose means it can't exit the channel anymore, which means it can't leave the cell. And phosphorylating again means that there are two phosphate groups on it, which destabilizes it and makes it favorable to split. And this is important because like it wants to be cleaved because it wants to be cut into two when it turns into pyruvate. And how the phosphate groups do this is through chemistry. If you've like taken like Chem 111 or especially 112, or like earlier this class when we talked about cell chemistry, you'll know that like if two ions are negatively charged, they won't want to make interactions with each other. They'll repel each other. So if there are two phosphate groups that are negatively charged on the glucose or fructose in this case, then like they'll repel each other. They want to split apart, and that's why this happens and why it gets phosphorylated twice. And these are two phosphate groups, or these are the two ATPs that are used invested into the glucose during investment. So ATP also serves as a negative regulator. So glycolysis doesn't need to happen if there's a high level of ATP. So the glucose has an active site and like an active site where like this like where the ATP normally binds in, and like one more site, a regulatory site or like an allosteric site. So at this allosteric site or this regulatory site, the affinity for ATP is like a lot lower than the active site. And so when ATP manages to bind into this regulatory site, it suppresses enzymatic activity, meaning that if ATP binds in, this cycle right here, this picture won't happen. So my question is, why do you think you need high levels of ATP to suppress its activity? So if there was like a low amount of ATP, like this would happen as is. But if there was a high level of ATP, then this won't happen because ATPs can like also serve as a negative regulator. So yeah, I'll give you like a few moments to think about that. But if not, I'll just like explain. Okay, so yeah, the reason for this is because even though like the affinity for like the regulatory site is lower than like the active site, if there was a high concentration of ATP, that means that like even if the affinity was lower because of like the higher concentration of ATP like randomly diffusing around and bumping in, it means there's a greater chance of like one of the ATPs being able to bind in to the allosteric site and then like causing it to suppress enzymatic activity. And so that's why like ATP can also serve as a negative regulator, even though like you need to invest ATP for SAP in the first place. So it's just a cool little thing that the cell does to make it more efficient. Where like because if you have like lots of ATP, you don't need to make more. So yeah, that's why it's like that. Write it down chat. Uh, yeah, I'll do that after I'm done recording. Yeah, just remind me. So moving on to more glycolysis again, the payout phase. So this starts once the glucose is split. So as you can see on the chart down here, there's a part where like it's like fructose 1,6 biphosphate, and it turns into like a dihoxy acetone phosphate, and that's like the part where it splits into two. And so this is like when the payout phase really starts. And so a phosphate that's not from ATP, it's from like another source, is added to each of the split glucose molecules, and this leads to the phosphorylation of ADP. And this is known as substrate level phosphorylation, where ADP acts as a substrate and ATP is a product. And this is well, that's why it's called substrate level phosphorylation, because ADP is the substrate. Where like in a lot of the other reactions we've learned about, or like if you take more bio classes later on, or like cell bio classes or like biochem classes, you'll learn that like usually ADP is a product. It's not it's not like used as a substrate. But this is why this is kind of like special and how or at least how I remembered it. And then so the second half of glycolysis occurs, since now there, or well, yeah, this second half of glycolysis occurs twice because now there are two molecules of glucose. And you should keep this in mind. It's kind of a recurring theme through this and like through like the Krebs cycle later on, where like technically everything we talk about like happens twice because it was split into two. So like one glucose molecule leads to like all these things happening twice. So another phosphate is added onto one of the glucose molecules after splitting, and this leads to an NAD plus molecule being reduced to NADH. So you can see that yeah, you can see that kind of here where it goes from like two into like the 1,3 biphospho disaccharide, or yeah, sorry, I can't pronounce that very well, but yeah. So it is special because NADH is a high energy molecule. It stores some of the energy from the original glucose molecule. And so the payout phase is when the invested ATP pays out. So you get more ATP back than what you put in. So remember like earlier, I said that like you invest 2 ATP molecules, you get out 4. So yeah, that's from the payout. You get more than like what you put in. And pyruvate is made in the process of glycolysis. And this is important. This is like kind of the main thing of glycolysis. Pyruvate is really the most important part of this. And we'll talk about more. We'll talk more about it later with like fermentation and stuff, where like glycolysis alone doesn't really net you that much energy. So it's really pyruvate where like when it goes through the Krebs cycle is like where you get the energy from. So next up, we have the citric acid cycle, or well, the entry into it. Use this another name for the Krebs cycle. So pyruvate is transported from the cytosol into the mitochondrial matrix. And oh yeah, glycolysis happens in the cytosol, by the way. Yeah, I probably should have mentioned that. But yeah, and then it enters the mitochondrial matrix and it's converted to acetyl coenzyme A, or CoA, or however you want to pronounce it. So you can see here where the pyruvate goes into the mitochondrion. And so acetyl coenzyme A is a molecule required for going into the citric acid cycle. So it's really important. Yeah, there's another, yeah, on the next page, there's a better chart that shows that. So in this process, a CO2 molecule and an NADH molecule is produced. As you can see here, the CO2 at stage 1 and at stage 2, NAD plus goes into NADH. And then, so like I said earlier, since glucose yields 2 pyruvate molecules, this means that this happens twice each time glycolysis occurs. So this means that there are 2 NADH molecules and also like 2 CO2 molecules here each time like a glucose is used. So the chart only shows like 1 pyruvate, but like there are 2 pyruvates because it was cleaved into 2 if you remember. So yeah, this happens twice for each glucose molecule. And then yeah, this happens in the mitochondria, which is the powerhouse of the cell. Just some quick background in case you don't know what a mitochondria is, or if you don't remember very well. Yeah, and the Krebs cycle only occurs in the matrix of the mitochondria. Yeah, and I add that because like I didn't know that much about mitochondria when I took 151 myself. Yeah, and then so we go into the citric acid cycle. So acetyl coenzyme A is fed into the cycle where 8 reactions occur, starting with acetyl coenzyme A and oxaloacetate making citrate. So you can see here where there's like pyruvate. It goes into it turns into acetyl coenzyme A. Then it goes into the Krebs cycle, and behind it is like the oxaloacetate, and it goes in, and like together they form citrate, which is like citric acid. And so in the process of this, not all at once, but like through each like some of these reactions, like what's reduced is 3 NADH molecules, 1 FADH molecule or 1 FADH2 molecule, 1 ATP through substrate level phosphorylation, and 2 CO2s. And if you look at the graph, you can kind of see it where like 3 reactions in, you get an NADH, then like after that you get another NADH, then you can just kind of read it and so forth, and you can see like where you get these molecules from. And so more energy is extracted from this process than used. So you get a net increase in energy. Yeah, I forgot the numbers exactly, but it's a lot more than what you get from glycolysis. Um, I don't know if your prompt that talks about it at all. I haven't looked at that yet, but yeah. And so more CO2 is created as a byproduct. So if you like took like an easier bio class like bio in high school, you'll know that like or like bio or like just general science classes anywhere, you'll know that like CO2 is a pretty widely known byproduct of cellular respiration. So this is kind of like how it works, where like you get CO2 from like as you make ATP and energy from the Krebs cycle. And so remember this is extracting energy from pyruvate, which originates from glucose. So this cycle is essentially here to get as much energy as possible from glucose. And Professor Francis used the term a lot of like potential energy. If you've like heard it before in like physics, it's used in physics a lot, where like glucose has a lot of potential energy, and like glycolysis happens, and then you create like pyruvate, and pyruvate by itself also has lots of potential energy, and like this Krebs cycle is essentially like exploiting that potential energy, and, like turning that potential into like real energy. And so this is why this cycle is important, and it's a good way to think about it too. Um, yeah. And then after this, we have the electron transport chain. So there are proteins embedded in the inner membrane of the mitochondria, or just like the inner membrane of the mitochondria, which are what facilitate the electron transport chain. So you see a complex 1, complex 2, complex 3, like that's those are proteins we're talking about. So NADH is created by glycolysis, and NADH is created by glycolysis in a citric acid cycle, and they're dumped into the electron transport chain. So in a series of oxidation and reduction reactions, the energy / electrons are passed down from protein to protein, like a staircase, which is a metaphor Professor Francis used. Yeah, I believe we talked about oxidation and reduction reactions too a lot in like Chem 112 or Gen Chem 2. And so in each step, energy is extracted from electrons to drive ATP production. So oxygen acts as the final acceptor of electrons in the electron transport chain. So if you think about it like in the metaphor, you would think about it as like being at the bottom of the staircase. So if you keep on going up staircase, you'll reach oxygen. And you also see it forming water here, H2O. Yeah. And this is important because like without oxygen, the electron transfer chain would just stop because there's nothing to dump electrons into without oxygen, which means that there would be no ATP production. And without ATP production, the cell would die because ATP is just needed as like energy. So my question is, why do you think oxygen would be a good choice to do this? Think about like the chemistry of oxygen and like what makes it unique and like how it interacts with electrons. And I'll like give it a moment or a few moments. Okay, yeah, that's fine. Yeah, when I took this class, electron transfer chain was not my forte, and I still don't like it. But yeah, the reason for this is oxygen is just really electronegative. So like if you learned like or we talked about it before, but basically it's like elements are closer to fluorine or more electronegative, and they're more willing to like accept electrons basically, which is why oxygen is really good for this. It's an organic molecule that's really electronegative, so it can accept electrons really well. And I'm not sure if we've talked about this that much before, but it's definitely a thing that comes up in chemistry a lot, or like orgo too if you haven't taken that yet. So yeah, that's why oxygen is a good choice to do this. And then get more on the electron transport chain. So there's energy is lost in each step of the electron transport chain, and this energy is used to do work. It pumps hydrogen ions into the intermembrane space against their gradient. So it's not just like randomly diffusing; it's like actively pumping like hydrogens, and so that requires energy because it's like normally they don't want to be in like intermembrane space, but they're pumping hydrogens into it, and that's why. So the flow of hydrogen ions from like an area of high concentration, which right now is intermembrane space, to an area of low concentration, which is the mitochondrial matrix, is what drives ATP synthesis. And I believe the metaphor Professor Francis used for this was like a water wheel and like I guess like a waterfall or like a river where like is flowing down, and the movement of like things flowing down like creates energy which can be then like used to do stuff. In the case of like the metaphor, it appears like electricity and stuff. In this case, it is what drives ATP synthase, which is the making of ATP using, or the making of ATP using energy provided by the electron transport chain. So hydrogen ions flow from the intermembrane space to the mitochondrial matrix, and this drives movement of the rotor on the ATP synthase. And then driving the rotor on the ATP synthase is that then drives movement of the ATPase portion of the ATP synthase, which then produces ATP from ADP, which is the like the substrate level phosphorylation. And so you get ATP from this basically. So yeah, that was a lot to take in, but yeah, hydrogen ions are pumped up, they flow down, the movement of it flowing back down then like drives ATP synthase and causes it to like make ATP from ADP. And that's basically the gist of it. Yeah. And so here, finally, we talk about fermentation. So this is the process of how cells make ATP. Oh, wait, hold on. I got a question. Intermembrane space is high concentration. Yeah, so the intermembrane space is where there's a high concentration of hydrogen ions, and it flows down to the mitochondrial matrix. So yeah, that's correct. Yeah. Okay, I don't know if I can go back right now. I'm clicking the button, but it's not letting me go back to my last slide. So, oh well. Yeah, if you have more questions, feel free to talk about it after class or after the review slide. Oh, here it is. So fermentation is a process of how cells make ATP from glycolysis alone when there is no oxygen around. So usually when fermentation happens, it's because there was no choice. So let's say if you couldn't breathe, for example, you couldn't get any oxygen in, then you would have to do fermentation because there's no oxygen around. So some organisms, such as humans, can only do fermentation for a short time. And this is because like a buildup of lactic acid is toxic. And lactic acid, if you don't know, is like a product that's formed. There's a little graph down here where you can see like the pyruvate, and it forms 2-lactate or like 2-ethanol too. And then the other organisms like budding yeast can survive off of fermentation if necessary too, but that's not us or most animals. And so basically what happens is only glycolysis is carried out since there's no oxygen. So extraction of energy from glucose is not as efficient as it otherwise could be. So yeah, remember that the citric acid cycle in electron transport chain was there to extract as much energy as possible from pyruvate. And this, if there was no oxygen, this like just wouldn't be possible. So I talked about earlier the potential energy of like glucose and pyruvate that like the citric acid cycle kind of exploits and uses, that if there wasn't oxygen, it wouldn't be able to do that. So this is way less efficient. So when fermentation happens, organisms need a way to dump electrons from NADH to reform NADH plus. So yeah, this is where fermentation comes in. It's a process of taking electrons from NADH and dumping them into pyruvate, which forms lactic acid and ethanol as waste products. So yeah, you can see the little charts down here where like glucose turns into pyruvate, then like if there was oxygen, it goes into like the mitochondrion and you see going through a cycle and like the electron transport chain. But if there was no oxygen, then it practices fermentation. And then yeah, that's it for my review session. Was there any questions that you guys wanted to ask? Go back to the electron transport chain. Yeah, sure. Do you have any questions about it? I remember also having a really hard time on the electron transport chain when I took this class. Thankfully, it was not on our exam, but yeah, I agree. I didn't like the electron transfer chain. It's a lot of information and it's really confusing too. The whole part about pushing against a gradient, I couldn't get that well either when I took the class, but yeah. Were there any like specific questions you guys wanted to ask? if you guys want to go over stuff on the problem set too. Oh, the first question. All right, yeah. Yeah, I'll do that while I'm here. So the first question, yep, was it this one where why do we need high ATP levels to suppress its activity? Yeah, so the glucose molecule has a binding site where ATP binds in, and this is what turns into like glucose 6-phosphate and like leads into glycolysis. But it also has an allosteric site, which if you don't remember from like earlier is like another binding site on the molecule, which in this case is glucose. And that allosteric site has a lower affinity for ATP. So normally, let's say if you have like 4 ATP or like not a lot of ATP, it would bind into like the normal site of the glucose and it would be able to like go through glycolysis. But the chances of it being able to bind into like the allosteric site when there's a low amount of ATP is like not high because its affinity isn't good. So like if you, I don't remember if that was 151 or 285 where we went over like KD and like affinity, but like if it's low affinity, that means that like when it bumps into each other, it doesn't stick together as long as they don't have like good affinity for each other. So if it was a low amount of ATP and like the chances of it being able to bind into the like allosteric site is really, really low. Like basically it's just not a thing. But then if you like increase the concentration of ATP to be really, really high, there'll be lots of ATP floating around. And then yeah, if you've talked about in like chemistry at all, where like you talk about like pressure and like lowering the volume or like increasing the concentration of ATP in this case, it would increase the likelihood of ATP being able to bind to the allosteric site, even though the affinity is lower. And then so that means that if there's a high concentration of ATP, it'll be more likely to bind into the allosteric site. And that means it'll be more likely to suppress um, enzymatic activity of glucose, or yeah, because ATP bind in. So the other site, do you mean the main binding site of like where ATP normally binds in? Oh yeah, so the allosteric site is there to regulate the regulate glycolysis essentially. Yeah, it's called the regulatory site. So if ATP is able to bind into the allosteric site, even though there's low affinity, that means that like that signals to the cell that there is a lot of ATP around because because it's low affinity, ATP will only be able to bind in when there's a lot of it around. So if it's able to bind in, that tells the cell that like there are lots of ATP around. And so if there's lots of ATP around, it also tells the cell that it doesn't want to make more ATP because it's already so much ATP around. So that's why if it's able to bind in, it suppresses activity. It's yeah. Sorry if that was confusing. If you want me to like go over it again or explain it a different way, let me know. Okay, yeah, cool. Okay, yeah, I'll stop the recording here.